Did you know that coral spawn can stay frozen for decades? Well, that's exactly what happened this year at Ningaloo Reef, but not in the open ocean, here at the Minderoo Exmouth Research Lab.
Most people don't get to see this.
Join our scientists for a behind-the-scenes look at what it takes to recreate the perfect ocean conditions, collect the spawn, and learn why cryopreserving this reef is so important.
So we're here standing in the darkness with red light because we are waiting for the corals to spawn. So in order for them to spawn, we need to make sure that we don't disturb them, and we do that with red light. So what we're looking for is retracted tentacles, and we're looking for that spawning bundle to be right at the surface of the coral. So it almost gives the coral a pimply look to it. And so that's something that we call setting, and that tells us that the corals should be ready to spawn very soon. So we're doing our little checks, our roving, to make sure that we'll catch them in the act. So over the last few weeks, these corals were collected across the Ningaloo, some up in Exmouth Gulf as well, and they were brought into the boat, and this is where we've been waiting for them for the last week, every night, doing a visual to prepare for the spawning. So once these corals spawn, we'll collect their little bundles, which has both eggs and sperm. We'll separate them out, and then we'll mix them in specific combinations. And over the next few days, we'll have them in our coral baby nursery at the back of the Minderoo Exmouth Research Lab. And in those specialized tanks, the baby corals will metamorphose, and they'll almost change, just like caterpillars change to butterflies. And they'll become little baby corals and then little teenager corals that then we'll work with in our experiments. Ningaloo has been under a lot of pressure this last year with the marine heat wave, which is very similar to having like a wildfire underwater. And so when seawater temperatures heat up, corals get really stressed, and we see sometimes big mass bleaching and mortality events. And unfortunately, this is what has happened all across the Ningaloo this last year. Over the last 50 years, about 50% of coral reefs have been lost around the world. And so we really need to be looking to help corals kind of get through the next few years while we bring our climate emissions under control and start to bring down water temperatures. This year, we're really excited about partnering with Taronga Zoo. And we've known that it's really important to cryopreserve or save genetic diversity before it's lost. And so we've been working for the last few years with DBCA and the Joint Management Body to try to get cryopreservation going with Ningaloo corals. And we were actually able to cryopreserve the first Ningaloo corals last night, which is a real success, and we're very happy for that. And we hope over the next few nights we'll be able to get more individuals, more species, so we can get them cryo-banked at Taronga Zoo where they can be safe for future generations in case the marine heat waves get even worse year after year.
We found just the one colony tonight that was set with gamete bundles ready to spawn. So isolated that coral. It's a little bit light. There are only a few bundles that it released, so the sample is a little bit lower concentration than ideal, but we'll still be able to work with it and get something out of it. So once the bundles break up, the eggs are nice and buoyant, full of lipids for the larvae, so they float to the surface, and then the sample we're after is at the bottom, so we can probably actually isolate that now. So we're just going to have a look at the sperm on the microscope here. We're going to activate it with caffeine, which will make sure that all the sperm are fully motile. And then we'll have a look at it on this software here, and that'll be able to tell us the number of sperm in the sample and the proportion that are swimming. We will prepare some cryovials, which are these tubes that we'll freeze the samples in. We'll combine it with the cryoprotectant, and then we'll cool it in liquid nitrogen using these devices here, and then transfer to our dry shipper. These samples, once we've cryopreserved them, put them into the dry shipper, we'll be taking them with us on the plane back over to Sydney. And they'll go into the Taronga Cryobiodiversity Bank. We're going to have a dedicated storage tank for all the samples that come from here in Western Australia, so that they can be kept together in their own location. And then those samples will be there for future use if they're needed. The cool thing about cryopreservation is that the samples that we freeze here are going to remain frozen but alive potentially for decades. And so what that means is that even if the coral colonies out on the reef are impacted, and even if we lose them out on the reef completely, we've still got access to the genetics from those corals, which means that we can use them to breed more corals in the future. And we don't lose that input from the genetic diversity. We've been biobanking coral samples from the Great Barrier Reef for over 10 years, but this is the first time we've ever actually come over to Western Australia and been able to start securing genetic diversity from these reefs here. And that's really, really important given the impact that we saw with the bleaching last year. So it's really great. It's a great first step to get over here and start banking some of that critical genetic diversity.
For this year's spawning treatment, for the first time we have corals from Exmouth Gulf. And this is pretty interesting because the corals in the Gulf are exposed to generally higher temperatures and also fluctuations in turbidity, so water murkiness, and higher salinity, nutrients, just an abundance of kind of more stressful conditions for corals. So the theory is that if these corals are used to being in stressful conditions, they may be more resilient to some of the conditions that will come with future climate change. And then hopefully if we spawn those with corals from Ningaloo, they may produce more thermally tolerant babies. So we're hoping to improve the strength and resilience of the next generation of corals on Ningaloo Reef with help from Exmouth Gulf. So Exmouth Gulf is a really incredibly diverse and important ecosystem as a whole. It's a huge body of water, but then also around that body of water, there's intricate intertidal habitats where varieties of mangroves, seagrass, samphire, and microbial mats are acting as huge carbon sinks, but also releasing a lot of nutrients and also particulate matter out into the Gulf. And a lot of people call that the lungs of Ningaloo. So basically these intertidal habitats are driving food webs that then go into the Gulf and over to Ningaloo, and also supporting a lot of that primary productivity, which then brings in your humpback whales when they come to rest, dugongs, and also supporting reef systems across the Gulf, which then could potentially help support Ningaloo into the future. So it'll be really interesting to see how these adult colonies respond to some of the thermal tolerance, so heat exposure experiments that we're going to expose them to. We're expecting the corals from the Gulf are going to fare a lot better. And we're also going to test them not only under increased heat, but also under increased light levels.
At long last, after 14 days of waiting, our colonies did finally spawn. The team worked tirelessly throughout the night to collect and cryopreserve as many as possible. This is because as our oceans change, so do the rhythms of life beneath the surface. And understanding these ecosystems is critical to protecting what comes next.